There's a shark on the sand about 100 feet below the surface, off the coast of San Diego. And you are looking straight at it and seeing nothing. That's not a figure of speech. The animal is right there in the frame. Brown, blotchy, about the length of your leg, folded into a crack in the rock with its tail tucked under its chin. The camera is pointed at it. The divers are close enough to touch it, and on the monitor it reads as one more patch of dead reef. Sand, algae, shadow, shark. All the same murky nothing. Then somebody screws a piece of yellow glass onto the front of the lens, and the rock turns into an animal. It happens all at once. A hard, electric green comes up out of the skin in a pattern nobody knew was there, spots and blotches, and a bright ladder of markings running down the flanks, glowing the way a highway sign glows when your headlights catch it at the wrong angle. The sand stays dead. The algae stays dead. Only the shark is lit. And it has been lit the entire time. That's the part that gets under your skin, if you let it. Nothing changed about the shark. Nothing changed about the water. The only thing that changed was a filter. For as long as people have been diving on that reef, this animal has been putting out a signal in plain view of anybody with the right eyes, and we have been swimming past it like it was a rock. One question we can already close. Does it happen? Yes. We can photograph it. We can measure the wavelengths. We have the chemistry now, down to the molecules, and I'll walk you through those because they're stranger than the glow itself. The open question is who it's for. A signal only counts as a signal when something out there is receiving it. Otherwise, it's a leftover, and an animal can absolutely produce a beautiful optical effect for no reason at all. The same way your kitchen faucet throws a rainbow on the wall in the morning without meaning anything by it. That's the case we're going to build here. Piece by piece, what was found, whether the thing is actually doing work, and who is on the other end of the line. Let's get the first misunderstanding out of the way, because almost every article you'll ever read about these sharks gets it wrong in the headline. And if you carry the wrong version into the rest of this, none of the argument will land. This shark does not make light. I mean that literally and mechanically. There is no light-producing organ anywhere on this animal. No chemistry burning fuel to throw photons into the dark. Put one of these sharks in a sealed black room, in true absolute darkness, and it disappears completely. No glow, no shimmer, nothing. What it does is convert. Blue light comes in from above. It hits molecules sitting in the pale patches of the animal's skin. Those molecules absorb it, hold that energy for a few billionths of a second, drop a small amount of it as heat, and release the rest as a photon with a longer wavelength than the one that came in. Blue in the front door, green out the back. The books call the drop in energy a Stokes shift, after a 19th century physicist who noticed the same trick in a lump of fluorite, which is where the word fluorescence comes from in the first place. A rock in a drawer in England, and now a shark on a reef in California. The important thing about that mechanism is the timing. It is instant, and it is dependent. Cut the blue light, and the green stops in the same instant. There is no reserve, no battery, no afterglow. The animal is a converter running on borrowed photons, and it is only ever as bright as whatever is falling on it. Now hold that next to the thing people usually confuse it with, because the contrast is what makes this worth spelling out. Bioluminescence is a chemical reaction. An organism combines a light-emitting compound with an enzyme, adds oxygen, and gets a photon out of the deal. That's real light manufacture. It works in a sealed cave with the door shut. It works at the bottom of the ocean, where no sunlight has ever arrived. And it works in sharks, which is exactly why the confusion is so easy to make. In 2021, a team led by Jerome Malafé published the first documentation of light production in the kitefin shark. working from specimens collected on New Zealand's Chatham Rise during a survey run by the country's National Institute of Water and Atmospheric Research. The kitefin gets to roughly 6 feet. That makes it the largest luminous vertebrate anybody has ever confirmed. Same paper covered 2 lantern sharks from the same waters. Their glow is concentrated on the belly, and the leading interpretation is counter-illumination, meaning the animal lights its own underside just enough to erase its silhouette against the faint light coming down from above. A shark that makes itself invisible by glowing. Deep sea logic. That's manufacture. What we're looking at on the reef off San Diego is conversion. 2 completely different problems with 2 completely different solutions. And the reason it matters so much for this particular case is that conversion has a hard constraint bolted to it. You can only convert what arrives. A bioluminescent animal is independent of its surroundings. It writes its own message. A fluorescent animal is a passenger. Its entire display is hostage to the light in the water at that moment, at that depth, at that hour, on that day. Take the blue away, and the message is gone. Which means that before we can talk about what the pattern says, we have to talk about whether there's enough blue down there to say it with. That's the whole physics problem, and we'll get to it in a minute, because it turns out the ocean is very, very good at delivering exactly the wavelength this system needs, and almost nothing else. One more piece of housekeeping while we're here, because somebody in the comments is already typing it. Yes, the jellyfish. The famous green fluorescent protein, the one that won the Nobel Prize in Chemistry in 2008 for Osamu Shimomura, Martin Chalfie, and Roger Tsien, the one that turned into the most useful lab tool in modern biology, came out of a jellyfish in the Pacific Northwest. And that jellyfish does both. It makes its own blue light chemically, and then a protein sitting right next to the reaction catches that blue and converts it to green. Manufacture feeding conversion? In the same animal, an inch apart. I bring it up because it sets an expectation that turns out to be wrong for the shark, and the way it's wrong is one of the better surprises in this story. For about 30 years, every time somebody found a fluorescent animal in the sea, the assumption was reasonable and obvious. Find the protein. Corals have fluorescent proteins. Eels have fluorescent proteins, evolved from a completely different family of molecules that normally shuttle fats around inside cells, which is its own small miracle of improvisation. So when chemists went looking inside the shark, they knew what they were hunting. They didn't find it. There is no fluorescent protein in this animal. What they found instead took years to pin down, and it belongs to a class of molecule that nobody in the field had connected to underwater light before. It's built from an amino acid you have in your own bloodstream right now. It's been chemically modified with an element that basically only shows up in marine chemistry. And when they tested it for something completely unrelated to vision, it did something that reframes the entire question of what this system is even for. That's coming. First, the water. Nobody set out to find a glowing shark. That's worth saying plainly because the tidy version of this story, the one where a team goes looking and finds what it was looking for, would be a lie. 2011. Little Cayman, in the Caribbean. John Sparks from the American Museum of Natural History and David Gruber, who splits his time between that museum and Baruch College at the City University of New York, are underwater at night doing something that sounds like a hobby and was actually assignment work. They're photographing corals under blue light for a museum exhibition about light-producing organisms. Corals fluoresce. Everybody in that world knows corals fluoresce. It's a known effect with a known audience, and they were there to document it for a display case. So the lights are on, the camera is running, the reef is doing its psychedelic thing, and an eel swims through the shot. Bright green, whole body, lit up like it had been dipped in something. They had no reason to expect that. Fluorescence in vertebrates was, at the time, basically a curiosity, a handful of records, Nothing anybody had organized into a question. And here's an animal with a backbone, in open water, in its own habitat, blazing green, in the middle of a frame that was supposed to be about coral. That's the whole origin. An accident inside somebody else's assignment. What happened afterward is what turned it into science. They went back with a plan. Blue lights, yellow filters, night after night across the Caribbean and then the South Pacific, pointing the rig at anything that held still long enough. 3 years later, in 2014, the group published a survey in the journal PLOS One that landed hard. 16 orders of fishes, 50 families, more than 100 genera, over 180 species putting out fluorescent light that nobody had cataloged, most of them living on reefs, most of them the kind of small, cryptic, patterned animals you'd never look at twice. The number kept climbing. By 2025, a team led by Emily Carr out of the same museum had pushed the confirmed count of fluorescent bony fishes past 450 species, with dozens of them documented for the first time in that single paper. They also mapped the trait onto the fish family tree and came out with an estimate that stops you for a second. The ability appears to have arisen independently more than 100 separate times, with the earliest origin they can place sitting somewhere around 112 million years ago, in the true eels, which is a nice loop back to the animal that started the whole thing. More than 100 independent origins. Evolution does not do that by accident, not at that rate, not across that many unrelated lineages. When a trait keeps reappearing in unrelated animals living in similar conditions, that's usually the signature of a problem being solved. Wings did that. Eyes did that. Whatever is going on with fluorescence in the sea, the ocean seems to keep asking for it. Sharks and rays came into the picture along the way, and they came in on the edges. As of the work that's been done so far, biofluorescence in that whole group, the cartilaginous fishes, is known from a small number of species belonging to 3 families that are only distantly related to each other. American round stingrays, wobbegongs, which are those flattened, fringed, carpet-looking sharks from the Western Pacific, and cat sharks, which is our animal. 3 families, scattered across the group, which means, again, independent origins, at least 3 of them inside the sharks and rays alone. And look at what those 3 families have in common, because it isn't taxonomy. Round stingrays lie buried in sand. Wobbegongs are camouflage specialists that spend their lives pretending to be a rug on the seafloor. Cat sharks are small, secretive, bottom-hugging animals that wedge themselves into rock and come out at night. Every one of them is an expert at not being seen. Animals that have invested everything in invisibility are, apparently, also the ones lit up like a scoreboard on a wavelength most of the neighborhood can't read. I find that combination very hard to explain as an accident, and it's the single strongest circumstantial argument in this entire case. Hold that thought, because it's going to have to survive some serious pushback later. Now, how does the trick actually get photographed? Because this is the part that people skip, and it's where a lot of the confusion lives. You need 2 pieces of glass, and they have to fight each other. Piece 1 is the excitation source, a lamp that throws blue, and only blue, into the water. You could do it with sunlight at depth, and eventually somebody will have to for reasons that'll become obvious. But for a controlled shot, you bring your own, because you want to know exactly what wavelengths went in. Piece 2 is the barrier filter, yellow glass on the front of the lens. Its job is to block the blue on the way back and let the longer wavelengths through. Without the barrier, you get nothing usable. The blue you're pouring into the scene is orders of magnitude stronger than the green coming off the animal, and it swamps the sensor completely. Everything reads blue. The shark's contribution vanishes into the flood the way a candle vanishes if you hold it up next to a stadium light. Put the yellow glass on and you delete the flood. What's left is the small amount of light that changed color on its way back, which is exactly and only the fluorescence. That's the setup. A blue fire hose and a yellow blindfold. And in between, an animal quietly doing chemistry. Divers who do this work wear the same thing on their masks. Yellow visors, blue torches. Somebody described the effect to a reporter as walking into a club where the only lighting is blue, and then one person steps onto the floor in an outfit painted with something that turns that blue into green. Everything else is a shade of the same color. That one person is a different color entirely. You couldn't miss them if you tried, which is a great image and a slightly dangerous one, because the yellow visor is doing an enormous amount of work in that comparison, and the shark does not have a yellow visor. Neither does anything else down there. That's the crack in the floor under this whole story, and we're going to walk right up to it. But first, the water, because the ocean has already done half of this animal's job for it, and understanding how it did that changes what the pattern on the skin is even competing against. Stand on a boat at noon and look down. The light going into that water is white, all of it. Every wavelength your eye can register, plus a pile on either side that it can't, all arriving together in roughly even proportions. That's what sunlight is. Now follow it down. The first thing to go is red. Take a red dive fin over the side and watch it. At the surface, it's red. At 20 feet, it's a dull, unconvincing red, like it's been left in the sun too long. By about 30 feet, it's brown. Keep going, and it turns a flat charcoal gray, and it stays that way for the rest of the dive. Bring it back up and it's red again, and it was red the entire time. Nothing happened to the fin. Something happened to the light. Water eats photons, and it's extremely picky about which ones. The reason sits in the water molecule itself. 2 hydrogens, 1 oxygen, bonds that stretch and bend at specific rates. Those motions have natural frequencies, and light in the infrared and deep red end of the spectrum happens to line up with the higher harmonics of those motions well enough to be absorbed and dumped as heat. Blue light doesn't line up nearly as well. So blue slips through while red gets converted into a tiny amount of warmth, about a body length below the surface. That's the whole reason the ocean is blue, incidentally, and it has almost nothing to do with the sky overhead. Pure water is faintly blue all by itself, in a bathtub, in a glass, if you have enough of it to look through. The sea just gives you a very long path length, so there's an order to the disappearance, and it's fairly consistent in clear water. Red is essentially gone within the first 30 feet or so. Orange gives out around 130. Yellow is finished before you reach 300 feet. Green hangs on longer, and blue keeps going. In clear, open ocean water, the wavelength that travels furthest before being absorbed sits right around 475 nanometers, which your eye reads as a deep, saturated blue. That's the champion. That's the one photon that gets to keep going after everything else has been stripped out of the beam. By around 330 feet down in clear tropical water, you're left with somewhere near 1% of the light that hit the surface, and almost all of what remains is packed into that narrow blue band. Keep descending, and it thins out from there. Somewhere around 3,000 feet, depending on who's measuring and how sensitive their instrument is, the last of the sunlight gives up entirely, and the ocean goes to black for the remaining however many thousands of feet there are beneath. Between those 2 points is the zone this story lives in, below the surface, where a full spectrum is still available. Above the true deep, where no sunlight arrives at all. The long blue basement in between. Now, what does that do to an animal living down there? It removes almost every tool that surface life uses to be seen. Think about how visual signals work up here. A cardinal is red because its feathers reflect red light and absorb the rest. A poison dart frog is yellow because it does the same trick at a different wavelength. Every color you've ever seen on an animal in daylight is that animal being selective about which parts of the incoming spectrum it sends back to your eye. It's a subtraction game. The sun hands you the full deck, and the animal decides which cards to show. Take away the deck, and the game ends. At 300 feet, there is no red light arriving, so there is no such thing as a red animal down there. You could paint a fish with the brightest cadmium red pigment ever manufactured, lower it to that depth, and it would be black, completely black. Not dark red, black. Because black is what you get when a surface absorbs everything that arrives. And there was only one thing arriving, which is exactly why so many deep-sea animals are red, by the way, and it's one of my favorite pieces of counterintuitive ocean logic. Red is the cheapest invisibility cloak in the sea. Deepwater shrimp, some of the mid-water fishes, that startling scarlet you see when a research vessel brings something up under deck lights. Down where they actually live, that scarlet is a hole in space, so the palette is gone. Everything at depth is some value of blue, running from bright blue near the top of that range to a blue so dark it stops feeling like a color at all. Brighter, dimmer, hazier, sharper, all blue. That's the world these sharks are trying to be seen in, or not seen in. And it's the reason a fluorescent animal is doing something structurally different from a colored one. Reflection can only ever hand back a portion of what it received. It's a budget with a hard ceiling. You cannot reflect more blue than arrived, and you cannot reflect a color that never showed up. Fluorescence walks around the whole problem. It takes photons at a wavelength that's abundant and hands them back at a wavelength that's scarce. It's a conversion, and in an environment with a single dominant wavelength coming in, conversion is the only way to be a different color than the water. Worth being precise about what that buys you, though. Nothing about fluorescence adds energy to the scene, and the amount coming back out is always less than went in, because you lose some to heat on every single conversion. Physics doesn't hand out free lunches. What you get is a shift in where that energy sits on the spectrum, and in a place with only one wavelength to speak of, that shift is worth more than raw brightness. There's a version of this you've walked past 100 times without thinking about it. Road crew vests, crossing guard jackets. That specific eye-hurting yellow-green they use. The color that looks somehow more saturated than a color should be able to look. That's fluorescence. Those fabrics are dyed with compounds that catch ultraviolet and short blue wavelengths from daylight, wavelengths your eye barely registers, and re-emit them right in the band where human vision is most sensitive. The result is a garment that appears to send back more light than falls on it. It doesn't, obviously. It's just moving photons out of a band you're bad at seeing and into the one you're best at. Same physics, same problem. The engineer who designed that vest was solving for a driver's eye at dusk on a wet highway, and evolution was solving for something in the dark at 300 feet, and both of them landed on the same move. But that vest works because human daylight vision is broad, and the fabric is loading up the exact band we're strongest in. For the shark, we now have to ask the harder version of that question, and it has two halves. Is there enough blue arriving at cat shark depth to run the conversion at all? And is green actually the right band to be moving those photons into, given what a shark's eye is built from? The first half has a clean answer. The second one is where this whole story gets complicated. Here's the clean half. There is plenty of blue. The swell sharks in the footage I described at the top were filmed in place on the Quine Art Desmeuchorbi on the wall of a submarine canyon off La Jolla at roughly 100 feet. That's shallow enough that a diver on a single tank can get there and back with time to work. At 100 feet in coastal California water, you've lost the red end entirely, and a good chunk of everything else. But the blue and blue-green coming down is more than sufficient to drive a fluorescent molecule. No question about it. You can watch it happen with the lamps switched off, using nothing but the daylight that made it down. That last point matters more than it sounds. A lot of the images that circulate from this work were made with blue torches because torches give you control and repeatability and a shot you can actually expose. But the effect does not require them. Natural light at that depth is already doing the job. The animal is already converting. The lamp is there for the camera. The shark was never waiting on it. The chain cat shark is a harder case, and it's worth being upfront about why. That species lives deeper. Records put it across a broad band, roughly 250 to 1,800 feet through most of its range, with the northern populations tending shallower and the southern ones going deeper still. Outer continental shelf, upper slope, rough rocky bottom, western Atlantic. Nobody is casually swimming down there with a camera rig, which means a great deal of what we know about that species under blue light came from animals in tanks and animals on lab benches, and that's a real limitation on the whole story that I'll come back to. At 1,800 feet, is there still enough blue to run the effect? Some, yes. The light is faint by then, but a fluorescent molecule doesn't have a switch-on threshold. It converts whatever it gets. At that depth, the conversion still happens. Whether the result is bright enough for anything to notice is a completely different problem, and one we'll have to be honest about. Now, the numbers on the light itself, because these are the numbers the whole argument hangs on. When you take these two sharks and hit their skin with blue, the light that comes back out peaks green. Specifically, the measured emission maximum in both species falls in a band around 525 to 535 nanometers. That's a strong, saturated green. If you saw it isolated on a screen, you'd call it the color of a laser pointer or an emergency exit sign. The researchers tested excitation at several wavelengths going in, 355 nanometers, which is ultraviolet, 390, which is that violet edge right at the limit of human sight, and 470, which is the honest one, because 470 is what's actually in the water. All three drive the effect. The relevant one is the last, and when the compounds were pulled out of the skin and measured directly in solution, the picture got broader. The extracted material absorbs across a wide range, wire, roughly 250 to 550 nanometers, and emits from about 300 out to 600. So it's a forgiving system, broad about what it will accept going in. A wide catcher's mitt with a green output. One detail from that work I keep turning over: under ultraviolet, the darker patches of skin put out a weak blue emission, around 455 nanometers. Different tissue, different chemistry, different color, sitting right next to the green. It doesn't do much for the animal at depth, since there's no ultraviolet down there worth mentioning. But it tells you the skin is chemically layered in a way the eye never suggests. So, blue in at 470, green out at 530. Call it a 60 nanometer shift. That's a big move. That's the difference between the color of the ocean and the color of a road sign. And that's where the second half of the question comes in. The one I said gets complicated. Is green the right place to put those photons? Because you can shift light anywhere you want, thermodynamically speaking. As long as you shift it downward in energy, the molecule could have landed on yellow, could have landed on orange. The chemistry allows a range, and we know it allows a range, because the same class of compounds in these animals comes in several versions with different spectral behavior. Something picked green, or something settled on green, and those are two very different stories. There's a complication I have to put on the table before we go further, and it cuts against the tidy version. The ocean is not uniformly blue. Clear, open ocean, far from land, deep water, no river runoff, very little suspended material. That water transmits best, right around 475 nanometers. Textbook blue. Coastal water is a different animal. Dissolved organic material from land, plankton, sediment, all of it absorbs at the short end and drags the transmission peak toward the green. In heavily productive coastal water, the wavelength that travels furthest can sit up in the 500s, in the yellow-green. Oceanographers have been sorting water into categories on exactly this basis since the middle of the last century. Now look at where the swell shark lives. Kelp beds off Southern California, some of the most biologically loaded coastal water on the west coast of North America. That is emphatically not the sterile blue of a mid-ocean gyre, which means the background light in a kelp forest at 100 feet is already shifted toward green. And an animal converting blue into green in that setting is producing a color the water is already producing on its own. The contrast you get in a photograph, where a yellow filter has surgically removed all the blue, is not the contrast a shark's eye would get with no filter at all in green-tinted water. Credit where it's due, because the people who did this work know that. The modeling in the original study explicitly ran the numbers across depths and found that the fluorescence increases luminosity contrast against the background and increases it between the light and dark patches on the animal's own skin in the conditions they modeled. The effect is real in the math. It's just smaller and more conditional than the photographs make it look. And the photographs are what everybody remembers. That gap between what the camera shows and what the animal could plausibly perceive is the central tension in this entire case. Every piece of evidence from here forward either narrows it or widens it. Before we get to the eye, though, we should probably meet the animals properly, because I've been calling them sharks, and I suspect the picture in your head is wrong in almost every dimension. Say the word shark, and a shape assembles itself in your head. Torpedo body, dorsal fin, teeth arranged for problems. Put that shape away. The swell shark is about the size and general attitude of a well-fed house cat that decided to become a fish. Adults run up to roughly 3.5 feet, and most of the ones you'd encounter are smaller than that. The body is soft-looking and heavy toward the front, tapering back to a tail that doesn't look built for speed, because it isn't. Brown and tan, mottled all over with dark saddles and pale spots, the exact color of a rock with algae growing on it, which is the entire point. The eyes are the thing that gets people. They're large, set high on the head, gold or green, with a vertical slit pupil that opens wide in the dark and narrows in daylight. Cat eyes. It's where the family gets its name, cat sharks. There are well over 100 species of them worldwide, and almost nobody outside of ichthyology can name one. They live along the eastern Pacific, from central California down through Baja and the coast of mainland Mexico, with a separate population way south off Chile. That's always been a little bit of a puzzle. Records put them from just below the surface down to about 1,500 feet, but that's the outer envelope. In practice, most of them sit between roughly 15 and 120 feet, on rocky, weedy bottom, and especially in kelp. Now the daily routine, because it's relevant to everything that follows. During the day, a swell shark does nothing at all. I mean that fairly literally. It finds a crack in the rock, or a cave, or the space under a ledge, and it goes in and stops. Divers regularly find them stacked in there like firewood, 2 or 3 or half a dozen animals piled on each other, motionless, tails folded under chins, gills working slow. No territoriality, no jostling, just a heap of sharks waiting out the daylight in the same hole. That's a social arrangement, whether or not anybody's called it one. Animals that pile up together are animals that tolerate each other, and animals that tolerate each other have to be able to tell each other apart from whatever else might be in the hole. File that away. At night, the shape unfolds and goes hunting, and the hunting style is patient and unhurried and, if I'm honest, a little bit sinister. The swell shark works slowly over the bottom and through the lower kelp, and a big part of what it eats is fish that are active during the day and torpid at night. Sleeping fish. It's been described in the literature as a specialization. The shark goes to work in the hours when its prey has effectively shut down and stopped responding to things. Barely a chase at all. More like harvesting. There's also the trick it's named for, and it's one of the best defensive moves in the ocean. When a swell shark is threatened, it bends its body into a U, reaches back, and takes its own tail fin in its mouth. Then it starts swallowing water. The stomach expands, the whole animal inflates, and it roughly doubles in girth in a few seconds. If it's already inside a crevice when it does this, the result is a shark that has physically wedged itself into the rock. You cannot pull it out. A predator can't get a purchase on it and can't fit a jaw around it. It just sits there, swollen and locked in, and waits. Do it at the surface, and it'll take in air instead of water, and there are consistent reports of the animal making a sound like a dog's bark when it forces that air back out. A barking shark. Nature does not care about our categories. Reproduction is the other piece that makes this animal feel less like a shark and more like something out of a folktale. Swell sharks lay eggs. Each one comes in a tough amber-green case, roughly the shape and size of a small purse, with long curling tendrils at the corners. The female wedges those tendrils around kelp stalks or sea fans, so the case stays put. Then she leaves, and the case sits there for 8 to 10 months. Think about that duration. Most of a year glued to a piece of kelp in a coastal ocean full of things that eat eggs, with a shark developing inside on nothing but the yolk it was packed with. No parent anywhere near it. When it finally hatches, what comes out is a few inches of fully formed miniature shark, patterned, functional, already capable of doing the U-shaped inflation trick if something bothers it. People find those cases washed up on California beaches all the time and call them mermaids' purses, which has to be one of the better common names anybody has come up with for anything. Put the whole profile together, and here's what you have. A small, slow, harmless, nocturnal, bottom-dwelling animal, camouflaged to disappear against a specific kind of rock, that spends its days packed motionless into a hole with other members of its own species, hunts sleeping fish by night, defends itself by becoming a plug, and abandons its offspring inside an armored envelope for the better part of a year. Nothing in that description predicts a light show. That's the collision at the heart of this. Everything about this animal's biology is an investment in not being noticed. The camouflage is excellent. The behavior is designed around stillness and concealment. And then you put a piece of yellow glass on a lens and the same animal is broadcasting a high contrast pattern that you could pick out from across the reef. An animal cannot be both maximally hidden and maximally visible in the same channel at the same time. So either the pattern isn't costing it anything, which would mean nothing is looking at that channel, or the pattern is buying it something worth the exposure. Either answer would be interesting. They lead to completely different biology. And before we can even start choosing between them, there's a second species to bring in, one that lives in conditions so different from the kelp forest that it functions as a natural experiment. The chain cat shark is smaller. Adults top out somewhere around 18 or 19 inches. You could hold one in two hands. And it's beautiful in a way that photographs badly, which is probably why nobody knows it exists. The base color is a warm tan, sometimes almost golden. Laid over that, in dark brown or black, is a pattern of interlocking lines that looks exactly like chain link fencing. Loops, links, chains, running the length of the body and out onto the dorsal fins. That's the name, chain cat shark. Somebody looked at it and saw hardware. It lives on the other side of the continent and a long way down, western Atlantic, from southern New England, along the shelf edge, past the Carolinas and Florida, and then a separate stretch in the northern Gulf of Mexico and down toward Nicaragua. The habitat is consistent wherever you find it, the outer continental shelf and the upper slope on rough, rocky bottom, in that band of ocean floor where the shelf stops being shallow and starts falling away. Depth is the key difference from the swell shark. Records run across a wide range depending on latitude, but the working band is roughly 250 to 1,800 feet. Northern populations tend to sit shallower and southern ones deeper, which is a pattern you see in a lot of temperate to subtropical species and usually tracks water temperature more than anything else. It lays eggs in cases like its Pacific cousin, and this is where it gets a little bit wonderful. Chain cat shark egg cases have been documented anchored to deep water corals down on the slope. The female threads the tendrils around the branches of a coral colony that may itself be centuries old and leaves the case there to develop in the cold and the dark. Nursery furniture built by an animal from a completely different phylum and standing longer than any human structure in the neighborhood. So now put the two species side by side, because the pairing is what makes this a scientific question instead of a photograph. Swell shark, Eastern Pacific, coastal, kelp, shallow, green-tinted productive water, active at night, diver accessible. Chain cat shark, Western Atlantic offshore, rocky slope, deep, clean blue water. Permanently dim, effectively unreachable without a boat and a net or a submersible. 2 ocean basins, 2 depth regimes, 2 water types, 2 entirely different ecological situations. Same green, same wavelength band, same family of molecule doing the work. That convergence is doing real argumentative labor, so it's worth being precise about what it can and can't support. These two animals are both cat sharks. They're in the same family, so a shared trait between them could just be inheritance, something a common ancestor had that both of them kept, which would say nothing about whether either one uses it. The stronger version of the argument comes from the wider picture, because within the sharks and rays as a whole, the fluorescent species we know about are spread across 3 families that are not close relatives. Cat sharks, wobbegongs, out in the Western Pacific, which are flat-fringed camouflaged ambush hunters that look like a piece of the seafloor with a mouth, and American round stingrays, which are small, disc-shaped, and spend most of their existence buried in sand with their eyes sticking out. Those 3 are separated by a very long time. Whatever's happening, it happened independently in each of them. And here's the part that got my attention when I first went through the literature. Look at what those 3 groups share behaviorally. Every one of them is a specialist in concealment: buried, flattened, wedged, mottled, nocturnal, cryptic. None of them is an open water schooling animal where everybody can see everybody all day long. Every one of them has committed, hard, to the strategy of being unfindable. An animal that has solved the problem of being invisible has created a new problem for itself. It's now invisible to everything, including the members of its own species it needs to find. That's a real bind, and it's a bind that has produced strange solutions all over the animal kingdom. It's the same pressure that gave fireflies their species-specific flash codes, and in some moths, a courtship song pitched up into the ultrasonic, quiet, and short range, in a world where the loudest thing overhead is a bat. When you've built your whole life around not being detected by one audience, you need a private channel to reach the other one. A private channel is exactly what a fluorescent pattern would be, if the conditions were right. It's invisible to anything whose eye can't separate that green from the surrounding blue. It only exists where blue light is the only light, which is to say in deep water, which is to say precisely where these animals live and know where a surface predator would be operating. It costs almost nothing to run, since the ocean is supplying the energy for free. On paper, it's a near-perfect design for a secret handshake, and the chain cat shark, specifically, resolves the complication I raised earlier about kelp forest water being green. Out on the upper slope, hundreds of feet down, well off the coast, the water is properly blue, clear, monochromatic, the textbook case. The species that lives in the best possible conditions for this effect is also the species with the strongest fluorescent patterning and, as we'll see, the only one of the two with a specialized skin structure that appears built to manage the light. That's an alignment worth taking seriously. It's also, on its own, worth almost nothing, because every word of the last few minutes has been about the light and the water and the animal's lifestyle, and not one word has been about the receiver. I've been arguing that the signal would be perfect if someone were listening. That's a design argument. And design arguments in biology have an ugly track record. Plenty of things look purpose-built and turn out to be structural accidents that nothing has ever used for anything. So we go to the eye. And this is where the study that started all of this did something quite unusual. The team went and measured shark vision directly, one photoreceptor at a time. The technique is called microspectrophotometry, and the description of it is more dramatic than the name. You start in the dark, total dark, or as close as the room allows, because light bleaches the very molecules you're trying to measure, and one careless flashlight ruins the sample. Working under infrared, which the tissue can't respond to, you take the retina out and tease it apart until you've got fragments small enough that individual cells lie separated on the slide. Then, you put one photoreceptor under a microscope and shine a beam of light through it that's narrower than the cell itself. One cell, one beam, and you sweep that beam across the spectrum, wavelength by wavelength, measuring how much light the cell swallows at each step. What you get out is a curve, a picture of exactly which photons this particular cell is good at catching and which ones sail straight through it. Do that a few dozen times across enough cells, and you have the animal's visual pigments directly measured. No inference, no behavioral guessing, no assuming that because a fish lives in blue water, it must see blue. Now, what's actually in there? Every photoreceptor in every vertebrate eye runs on the same basic part, a large protein called an opsin, folded through the cell membrane, and cradled inside it a small molecule derived from vitamin A. That small molecule is the light catcher. When a photon of the right energy hits it, it snaps from one shape into another, and that shape change kicks off a cascade that ends with the cell changing its electrical output. That's vision, one molecule bending. Everything else is bookkeeping. The clever part is the protein around it. The amino acids lining the pocket where the light catcher sits determine which wavelengths it's most likely to grab. Swap a handful of those amino acids over evolutionary time, and you slide the whole sensitivity curve up or down the spectrum. That's how a deep-sea fish ends up tuned to blue, and a desert lizard ends up tuned to red, using the same fundamental part. The peak of that curve has a name: lambda max, the wavelength the cell is best at. For the swell shark, the number came back at 484 nanometers, give or take about 3. The cell keeps working on either side of that, falling off in both directions, still catching a useful fraction of what arrives anywhere between about 440 and 540. For the chain cat shark, 488, give or take 3. Same working range. Look at where those numbers sit: 484, 488. And the wavelength that penetrates clear seawater furthest is right around 475. That's not a coincidence, and nobody in the field would treat it as one. It's one of the most repeated results in the study of marine vision. Animals that live in blue water evolve eyes tuned to blue water again and again, in lineages with no common ancestry to speak of. The eye follows the light. So far, so tidy. The shark is tuned to the ocean it lives in. Then comes the result that complicates everything. When the researchers went through those retinas, they were looking for two kinds of cell: rods and cones. You have both. Almost every vertebrate has both, in some ratio. Rods are the dim light machinery, big, sensitive, slow to respond, and wired in groups so that dozens of them can pool their signals into one output line. That pooling is what lets you see anything at all on a moonless night. The cost is resolution and speed, and the fact that they max out and stop reporting once the light gets strong. Cones are the daylight machinery, less sensitive, faster, wired more directly, And this is the part that matters. They come in multiple types with different lambda max values. You have three: a short wavelength type, a middle, and a long. Color for you is your brain comparing the outputs of those three against each other. In the swell shark and the chain cat shark, the retina came back rods, just rods. No cone population to speak of. One class of photoreceptor, one visual pigment, one channel. And a single channel visual system cannot see color. That's a hard result. And it comes out of a principle that took physiologists a long time to accept, so it's worth doing properly. A photoreceptor does not report wavelength. It reports a count. When a photon is absorbed, the cell responds. The response is identical no matter which photon did it. A blue photon that gets absorbed and a green photon that gets absorbed produce exactly the same event inside that cell. The wavelength information is destroyed at the moment of capture. All the cell can tell you afterward is how many hits it took, which means a dim light at the cell's favorite wavelength and a bright light at a wavelength it's bad at can produce the identical signal. The cell has no way to separate them. Physiologists call this the principle of univariance, and it's the reason color vision is impossible with one receptor type. You need at least two with different tuning and a nervous system that compares them. The comparison is the color. One channel gives you brightness. That's the whole menu. So here's where the case stands at this point, and it's not comfortable. The shark is putting out green light. The shark's eye is tuned to blue. And the shark's eye is physically incapable of registering green as a different color from blue, because it has no second channel to compare against. From the animal's side, there is no green. Which sounds like it should end the investigation right here. The signal exists, the receiver exists, and the receiver has no way to read the message, except that framing contains a mistake, and it's a mistake almost everybody makes the first time through, including me. I spent a while assuming it was fatal. It isn't. And the reason it isn't turns on something the yellow filter has been hiding from us this entire time. The mistake is assuming a signal has to be a color. Take a black and white photograph. No color information whatsoever. A single channel, brightness only. Exactly the situation the shark is in. And you can read a face in it. Read an expression. Read the weather, the time of day, the material of somebody's coat. Ansel Adams built a career in that channel, and nobody ever complained it was missing something. Text on a page is one channel. Chess is one channel. Radar is one channel. Enormous amounts of information move through pure luminance every day without any color involved at all. Whether the shark sees green as green has a settled answer. It doesn't, and it can't. The live question is whether the fluorescence changes how bright things look, and that's a numbers problem with a real answer. Go back to the rod. Lambda max at 484 nanometers for the swell shark. Still responding at reduced efficiency all the way out to about 540. The fluorescent emission peaks around 530, which is inside the window, toward the far edge of it, on the downslope, but inside. The shark's photoreceptors absorb that green light. They just don't label it. Every green photon that lands gets counted as one more unit of brightness, thrown into the same bucket as the blue. That changes the whole calculation, because now the pale patch on a shark's flank has two sources of light coming off it. It reflects some fraction of the ambient blue, like any surface would, and it emits green on top of that, converted from blue that would otherwise have been absorbed and lost. Two contributions, one bucket. The patch returns more photons to a nearby rod than it would if the fluorescence weren't happening. That's a brightness increase, real, physical, measurable, and available to a colorblind eye. Now the honest discount, because it's substantial. A green photon at 530 nanometers is worth less to that rod than a blue photon at 480. The absorption curve has fallen off by then. You're catching a reduced fraction of what arrives. So the fluorescence contributes to brightness at a penalty, and any calculation that ignores that penalty is cooking the books. Set against that, there's a second effect that runs the other way, and it's the one I find most persuasive in the entire physical argument. The dark patches don't fluoresce. The chemistry that produces the green is confined to the light-toned regions of the skin. The dark markings, the saddles on the swell shark, the chain link lines on the chain cat shark have none of it. They absorb light and hand back very little. So the contrast the shark's eye is being offered is not between a glowing animal and a dim ocean. It's internal. It's between one patch of the same animal that reflects and emits, and another patch inches away that does neither. Bright next to dark, with the gap widened by the fluorescence rather than created by it. Internal contrast is how patterns work. A zebra's stripes work on the difference across each boundary. Absolute brightness barely enters into it. The modeling in the 2016 study ran exactly this calculation, across a range of depths, using the measured absorbance curves from the animal's own photoreceptors and the measured emission from their own skin. The finding was that fluorescence increases luminosity contrast with the surrounding water and increases the contrast between light and dark patches on the animal itself throughout the depth range they tested, and there's a trend inside that result which is the most interesting thing in the paper. The deeper you go, the more the fluorescence matters. Think about why. As you descend, the ambient blue thins out. Less light arriving means less reflected light coming off every surface, including the shark. But the fluorescent contribution scales with the blue too, and it's arriving in a band where the background water is comparatively dark, because the water is not making green on its own down there. So the emitted component becomes a larger and larger share of the total signal leaving the animal, relative to a background that's fading toward nothing. At shallow depth, the fluorescence is a small addition to a bright scene. Go down far enough and it's most of what distinguishes the animal from the rock. That's the argument for why a deep-living, dim-water species is exactly where you'd expect this to be worth something. And it's why the chain cat shark, sitting out there on the slope in the dark in properly blue water, is the better test case, even though it's the harder one to observe. So we come back to the camera. The images that made this famous, the neon green sharks, are made with a filter that deletes blue completely. That image is a lie of omission, accurate as a map of where the fluorophores are, wildly misleading as a picture of anything's experience. So they built a second camera. The rig was a Red Epic Cinema camera with a 24mm lens and a custom stack of filters on the front, and the stack was specified from the photoreceptor data. A Raton 44, a gelatin filter, a 575 nanometer short pass, and a 400 nanometer long pass, layered to approximate the spectral response the researchers had measured coming out of the cat shark's own retina. This one wasn't built to make the fluorescence look good. It weighs every wavelength roughly the way the shark's rods weigh it, then adds them all into one number the way a single-channel eye has to. They called it the Shark Eye camera, and they took it down into the canyon. What comes back on that camera is worth describing carefully, because it's the closest thing anybody has to the animal's own point of view, and it is not a rave. It's dim. It's monochrome. And the pattern is there, muted, grainy, low contrast. A ghost of the pattern rather than a billboard, but visible, holding its shape against the background, and getting relatively more distinct as the depth increases and everything else fades. I'll say plainly that those images convinced me far more than the pretty ones did, and for exactly the reason you'd expect. They're underwhelming. Nobody makes an underwhelming image on purpose to sell a hypothesis. That footage looks like what a modest, real, physical effect ought to look like, and it survives the removal of the thing that was doing all the work in the publicity photos. So the physics holds up, with caveats. The animal is producing extra photons. The eye can absorb them. The contrast improves with depth. There's still no proof anyone's paying attention, but there's now a second piece of evidence pointing the same direction, and it comes from a completely different kind of observation. Because when you photograph a lot of these sharks and you start sorting the pictures, a pattern shows up in the pattern. The males and the females don't match. Sort the photographs by sex, and the picture changes. On a female swell shark, there's an arrangement of bright fluorescent spots across the head and around the face, a mask, distinct enough that it reads as a feature and not as noise, sitting in a specific place on a specific part of the body. Underneath, along the belly, she carries a denser scatter of fluorescent spotting than the males do. Males don't have the mask. That's the observation, and it's a simple one. And simple observations of this kind have started more arguments in biology than almost anything else. Then there are the claspers, and this is the detail that made everybody sit up. If you've never looked closely at a shark, here's the anatomy. Male sharks and rays have a pair of structures formed from the inner edges of the pelvic fins, stiffened with cartilage, rolled into grooved tubes. They're called claspers. They're the organs of internal fertilization, and only males have them. It's one of the oldest reproductive systems on Earth. Sharks were doing internal fertilization a couple of 100 million years before mammals worked it out. In both of these species, the claspers fluoresce strongly. Take that in as an anatomical fact for a second. Actually, let me put it more plainly than that. Out of all the surface area on a shark's body, the part that lights up most intensely is a structure that exists for one purpose and that only one sex possesses. If somebody handed you that observation cold, with no other information, you'd guess sexual signaling on the spot. It's the most obvious reading available, which is exactly why it needs handling with tongs, and we'll get to that. The chain cat shark shows its own version. The dimorphism there is in the reticulated pattern itself, that chain-link tracery, which comes out differently between the sexes when you image it under blue light. Females carry a more pronounced version of the network. Males carry the strongly fluorescent claspers. And underneath the fluorescence, in that species, there's a second layer of sexual difference that has nothing to do with light at all. The skin itself is built differently in males and females. Shark skin is covered in dermal denticles, which we'll come to properly in a bit. And if you count them under a microscope, you find that the density varies by body region and by sex. Work on the chain cat shark has counted them across the back, the belly, the flanks, and the front of the dorsal surface, and the numbers land in a broad band, running from roughly 100 per square millimeter at the sparse end up past 500 in the densest regions, with males tending to sit toward the higher end in most of the areas measured. The shape differs too. The crowns, the ridges, the way they overlap. Now, why would a male and a female shark need different skin? The answer is not romantic. Cat-shark mating involves biting. The male grips the female, usually around the pectoral fin or the flank or the gill region, and holds on with his teeth for the duration. It's a physical process, and the female takes the wear. Across several cat-shark species, females have been found to carry heavier denticle armor over the regions that get bitten, and the leading interpretation in that literature is straightforward protection, thicker skin where the teeth go. I mention this now, well before we need it, because it sets up a trap that I think a lot of the popular coverage of this story walked straight into. Here's the shape of the trap. The fluorescent pattern is sexually dimorphic. Everybody agrees. That's what the photographs show. But the skin is also sexually dimorphic for entirely mechanical reasons that have nothing to do with light. And if the skin structures are involved in producing or shaping the fluorescent appearance, which we're going to see that they are, then a difference in fluorescent pattern between males and females might be a downstream consequence of a difference in armor. Two sexes, different skin, built for different jobs during mating, and the optical difference falls out of the structural one for free. That would produce exactly the observation we have: a sexually dimorphic glow generated by an adaptation that was never about glowing. I don't think that's the whole story, and I'll lay out why later. But it's a live alternative. It's biologically reasonable. And any argument that jumps from the pattern differs by sex to the pattern is a mating signal has skipped a step that matters. Set that aside for a moment, though, and look at what sexual dimorphism in a visual character usually means when it does turn out to be real. Because the logic there is well established and it's worth having in hand. When two sexes of the same species look different from each other in some visible way, and the difference isn't explained by size or mechanics or diet, the standard explanation is that the difference is being read by somebody, usually by the other sex. Peacock tails, cardinal plumage, the elaborate structural blues on a bird of paradise. These traits are expensive, they're sexually restricted, and they exist because a preference on the other side kept selecting for them. The test has nothing to do with whether the trait looks impressive to us. What counts is whether the receiver's sensory system can detect it, whether the trait varies in a way that carries information, and whether behavior actually changes in response. We've got the first one. The cat shark eye can detect a brightness difference in that band. Established, measured, done. The second one is partially in hand. The trait does vary, systematically, by sex, in a consistent location on the body. The third one is missing entirely. Nobody has ever watched a cat shark respond to another cat shark's fluorescence. Not in a tank, not on the seafloor, not on video. No approach test, no preference trial, no courtship sequence recorded under natural blue light with the fluorescence manipulated and the animal's reaction measured. That's the hole in the middle of this case, and it has been sitting there, unfilled. Since the first paper came out, everything we've built so far is a chain of physical plausibility. Strong physical plausibility, measured at every link. But plausibility is what you have before you have an answer. So let's go the other way for a while and ask what the glow actually is. Because when chemists finally cracked that open, the answer had almost nothing to do with what anyone had been expecting, and it dropped a completely different hypothesis onto the table. 2019. 3 years after the vision work, and the question of what was actually producing the light had been sitting open the whole time, Gruber went and got a chemist, specifically Jason Crawford at Yale, whose lab spends its time on small molecules, the little stuff that organisms make in the course of doing metabolism. The compounds that get built, used, converted, and taken apart again, often in tiny quantities, often for reasons nobody has worked out. The first author on the paper that came out of it was Hyun Bong Park. James Weaver, over at Harvard, handled a lot of the structural imaging. Vincent Peerybone, who had been on the fish fluorescence work from early on, was in there too. That collaboration is the reason this got solved, and the reason is a little bit embarrassing for the field. For about 30 years, everybody looking for the source of fluorescence in a marine animal was looking for a protein. The reason is Equirria, the jellyfish, and the green fluorescent protein that came out of it and went on to remake cell biology. It worked. It kept working. Corals turned out to be full of fluorescent proteins. When the eels were finally worked out, they had proteins too, and beautifully weird ones evolved from a family of molecules whose ordinary day job is ferrying fatty acids around inside a cell. Some ancestral eel took a fat-handling protein and turned it into a lamp. So the search image was set. Find the protein, sequence the transcript, clone it, express it, characterize it. Done. Nobody found a protein in the shark. Years of it. That kind of persistent failure usually means the search image is wrong, and it took bringing in somebody who thinks about metabolites for a living to change the question. Here's how they actually ran it, and I like this part because the logic is so clean you could explain it to a kid. Step 1. Find out where the light is coming from. They put shark skin under fluorescence microscopy at a range of magnifications, working from the whole patch down toward individual structures. The green resolved into the pale regions of the skin, and within those, into specific locations. Step 2, and this is the move that cracks it. Take one animal. Cut out a piece of light skin. Cut out a piece of dark skin. Same shark, same day, same everything, except the tone. Grind both up. Extract both. Run both through the analytical machinery side by side. Now compare the two lists. Whatever appears in the light skin and is missing from the dark skin is your suspect. Everything else, every compound the two samples share, is background. Blood, muscle, structural protein, ordinary cellular housekeeping. All of it cancels out because it's in both columns. That's subtractive chemistry, and it turns an impossible problem into a manageable one. The search space collapses down to the difference between two pieces of one animal. What fell out of that comparison was a family of small molecules that nobody had ever described, present in the light skin, absent or down at a trace in the dark. Then comes the grind, mass spectrometry to get the exact masses and the fragmentation patterns, nuclear magnetic resonance to work out how the atoms are actually arranged, piece by piece, until you can draw the structure on a whiteboard and defend every bond in it. And what they drew was not a protein, nothing close. The scale difference here is worth a second, because it changes how you should picture the whole system. Green fluorescent protein is a machine. 238 amino acids folded into a barrel, with the light-emitting part sitting protected in the middle of it, like a light bulb inside a lampshade. The cell has to transcribe a gene, translate it, fold the chain correctly, and let the internal chemistry mature before any light comes out. It's a substantial investment. What's in the shark is a few dozen atoms, small molecules, the kind of thing an enzyme makes in 1 or 2 steps from something the animal already had lying around. Different engineering entirely, same output. And the something the animal already had lying around turns out to be an amino acid you're carrying right now, in whatever quantity your last meal supplied. Tryptophan. It's the biggest of the 20 standard amino acids that build proteins, and the most structurally elaborate. Most amino acid side chains are modest little things. Tryptophan's is a double ring, fused, flat, and electron-rich, and that ring system is what makes the whole story possible, because rings like that interact with light. You've encountered tryptophan in 100 contexts without noticing. It's the one people blame for post-Thanksgiving drowsiness, which is mostly a myth built on real chemistry, since tryptophan is the raw material your body converts into serotonin, and then into melatonin. It's also the starting point for niacin. It's in turkey, and it's in cheese and eggs and oats, and it's in you. And it fluoresces, all by itself, in a test tube. No modification required. Shine ultraviolet at around 280 nanometers into a solution of pure tryptophan, and it glows back at you just past the edge of human sight, up around 350 nanometers. That property is so reliable that biochemists use it as a standard tool. You can track a protein's folding by watching how its tryptophan residues change their glow as the structure moves around them. So the raw material was never exotic. Every animal on Earth has it. The fluorescence potential was sitting there in a common amino acid the entire time. The question is what the shark does to it. And the answer involves an element that almost never shows up in the chemistry of land animals, that's sitting in enormous quantities in every liter of seawater. And that turns a faint violet glow into a bright green one, bromine. There's roughly 65 milligrams of bromide dissolved in every liter of seawater. That's a small number next to the chloride, which runs about 300 times higher and is most of what makes the ocean salty. But bromide is everywhere in the sea, in every liter, forever. And any organism living in it has a permanent, unlimited supply of an element that land animals almost never touch. Marine chemistry is full of the consequences. Sponges, algae, sea slugs, tunicates, bacteria living in sediment. Pull a novel compound out of a marine organism, and there's a real chance it has a bromine atom stapled onto it somewhere. Chemists who work on marine natural products expect it the way you'd expect rust on something left outside. The most famous example in human history is a color, Tyrian purple, the dye of the Phoenician coast, produced from the glandular secretions of predatory sea snails, in quantities so miserably small that it took thousands of animals to dye a single garment. Roman law eventually restricted the deepest shade of it to the emperor. There were sumptuary rules about who could wear how much. For centuries, it was worth more by weight than gold, and the entire industry ran on rotting shellfish in vats outside city walls, because the smell was reportedly unbearable. The molecule is called dibromoindigo, two bromine atoms attached to a double ring system in a marine animal. The rings the shark is working with are the same basic architecture, which means the purple on a Roman emperor's shoulder and the green on a cat shark's flank are, at the level of the chemistry, distant relatives. Both are what happens when a marine animal gets its hands on bromine and starts decorating a ring. I find that connection delightful, and it's worth not overselling. Different molecules, different pathways, different purposes. What they share is a chemical logic. In the sea, bromination is a normal move, and bromination changes what a ring system does with light, which is the whole point. Plain tryptophan glows in the near ultraviolet. Invisible to you, invisible at depth, useless as a signal in the ocean. The compounds in the shark's skin glow bright green, out around 530 nanometers. That's an enormous shift in the emission, somewhere near 180 nanometers down the spectrum, and it's the difference between a molecule that does nothing visible and a molecule that lights an animal up. Whatever the shark's biochemistry is doing to that starting material, that's the distance it's moving it. Now the pathway, and this is where it stops being a story about sharks. When your body has tryptophan, it doesn't need for building proteins. It takes it apart. The main route it uses for that is called the kynurenine pathway, and the great majority of the surplus tryptophan in a vertebrate goes down it. Enzymes crack open the ring, and the fragments get processed through a series of intermediates with names like kynurenine, kynurenic acid, and quinolinic acid, eventually feeding into the production of a molecule your cells cannot run without. This is not obscure biochemistry. It's happening in you continuously. It has been happening in vertebrates for hundreds of millions of years, which is why it's present, more or less intact, in a shark. And in vertebrates generally, the intermediates in that pathway are not inert. Several of them are biologically active in ways that have kept pharmacologists busy for decades. Some interact with receptors in the central nervous system. Others are tied into immune regulation, with the first enzyme in the chain ramping up when the body is fighting an infection. It's a disposal route that doubles as a signaling system. So here's what the shark appears to be doing. It takes tryptophan. It brominates it, using an element that only the ocean supplies in quantity. And then it runs the brominated version down a metabolic pathway that every vertebrate already has, that was already there doing something else, that has been in the family since long before there were sharks. What comes out the other end is a collection of brominated kynurenine-related compounds with different spectral behavior, concentrated in the pale patches of the skin, absent from the dark ones. A whole family of them. That detail cuts both ways, and both edges are sharp. Read it one way, and it's exactly what you'd expect from a metabolic pathway simply running. Feed a modified substrate into an existing assembly line, and you get a spread of related products. Because that's what assembly lines do. They produce a distribution. Under that reading, the green is a consequence of chemistry the animal is doing for entirely different reasons. And the color is decoration on a process that was never about color. Read it the other way. And a family of compounds with different spectral properties is a palette. It's the raw material for tuning. If you needed to shift the emission or produce different shades in different body regions, or match a particular water type, having a suite of related fluorophores is precisely what you'd want in the cabinet. Same observation, two completely opposite conclusions. Welcome to Inference in Biology. There's an everyday version of the first reading sitting in the door of somebody's refrigerator right now. Tonic water glows. Take a bottle out to a bar with a black light, and it lights up a ghostly pale blue, brighter than anything else on the table. That's quinine, which is in the drink because it's bitter, and it's in the drink historically because British colonial officials in India were taking it as an anti-malarial and needed something to make it drinkable. The gin came along to make the quinine bearable, incidentally, which is the opposite of how most people assume that drink got assembled. Nobody put quinine in tonic water to make it fluoresce. The fluorescence is a property the molecule happens to have. It has been sitting in that bottle, unnoticed and unused, for 2 centuries, doing absolutely nothing for anybody, until somebody pointed a black light at it. That is a real and serious model for what might be happening in this shark. A molecule that's present for one reason, carrying an optical property that costs nothing and does nothing. To argue against it, you need something the tonic water doesn't have. You need evidence that the animal is doing extra work to make the light better, which is exactly what the next piece of the story provides. And it lives in the strangest tissue on a shark's body. Run your hand along a shark from head to tail and it feels like wet rubber. Run it the other way, and it takes the skin off your palm. That's the tissue we're going to talk about, and it's one of the truly alien things about this whole lineage. A shark is not covered in scales the way a bass or a salmon is. Bony fish scales are thin overlapping plates of bone that grow outward from the skin and get bigger as the animal gets bigger. With growth rings you can count like a tree. Shark skin is covered in teeth. I'm being literal. Dermal denticles, sometimes called placoid scales, and every single one is built the way a tooth is built. There's a central cavity with a blood supply and nerve tissue. Around that dentine, over the outside, a hard enameloid layer. Underneath, a base plate anchoring the whole thing into the dermis. Take one out, section it, put it under a microscope next to a cross-section of a tooth, and you'd have trouble telling them apart. Same developmental machinery, same instruction set, expressed in the skin. The relationship between the two runs deep enough that there's a long-standing idea in the field that shark teeth and shark skin denticles are variations on one ancient structure, and that the jaw's teeth are essentially skin teeth that ended up in the right place. Whether that's exactly how it happened is still argued, but the family resemblance is undeniable at the tissue level. So a shark is wearing a coat of thousands of tiny teeth, all pointing backward, which is why the skin feels smooth in one direction and like 80 grit in the other. That texture had a name and a market long before anybody understood the biology. Shagreen. Woodworkers and cabinet makers use dried shark skin as an abrasive for finishing. And it works because it is functionally a sheet of sandpaper that grew itself. Japanese swordsmiths wrapped hilts in ray skin, which is the same tissue, so the grip wouldn't slip when a hand got wet. The denticles don't grow. That's another difference from fish scales. Each one comes in at a fixed size, and as the shark gets larger, it adds more of them and sheds old ones. The armor is replaced piecemeal, continuously, for life. Now, what are they actually for? Protection, obviously. Thousands of enamel-coated points make a difficult surface to bite. But the more interesting answer is water. Look at a denticle crown under high magnification, and it isn't smooth. It carries a set of fine parallel ridges running front to back, like the ribs on corduroy, spaced in the range of tens to a couple hundred microns, depending on the species and the body region. Those ridges are called riblets. and they interact with the thin layer of water dragging along the shark's surface. A fast-moving surface in water develops a boundary layer, and in that layer the flow can break into small turbulent swirls that eat energy. The riblet geometry Benayas appears to organize that flow, keeping the disruptive vortices lifted slightly off the surface and reducing the drag penalty. Engineers have been chasing this for decades. It fed into swimsuit fabrics, into research on aircraft skins, into coatings for ship hulls. The physics is real even where the commercial claims got ahead of the data. And then there's the property that becomes important later. So pay attention to this one. Denticles resist fouling. Put a smooth, clean object into seawater, and it starts getting colonized almost immediately. Bacteria land within hours and lay down a film, then algae, then things with shells. Anybody who owns a boat pays for this problem twice a year. Shark skin doesn't do that. The microscopic texture, the spacing, the ridges, the overlapping arrangement, all of it makes a lousy landing site for organisms trying to settle and attach. There's an entire branch of biomimetic materials science built on copying that geometry onto surfaces where you want to prevent biofilm, including hospital surfaces. So denticles are armor, and they're hydrodynamics, and they're an anti-fouling system. 3 jobs. The 2019 team went looking for a 4th. They put cat shark skin through the kind of imaging you use when you want to know what a structure is actually made of and how light behaves inside it. Fluorescence microscopy, across scales, structural analysis, the whole apparatus. And they found that in one of the 2 species, the denticles are doing something with the light. In the chain cat shark, the denticles are not just sitting on top of a fluorescent surface. They're participating. Some of them behave as optical waveguides, taking the fluorescent light and channeling it along their own length, guiding it out from the skin along the axis of the structure. The description that ended up in the coverage was lightsabers, which is exactly as ridiculous and exactly as accurate as it sounds. That's a fiber optic, functionally. A structure that captures light at one end and conducts it along a path, rather than letting it scatter in every direction at once. Your internet runs on the same principle at a much larger scale and with much better materials. And the chain cat shark doesn't have one kind of denticle. It has 2. Smaller ones that do the light guiding, and larger ones that don't. 2 structural classes, one of which is optically active, distributed across the skin of an animal whose pale patches are loaded with fluorescent compounds and whose dark patches have none. Now go back to the swell shark, and here's where the comparison earns its keep. The swell shark's denticles don't do it. Same family, same green chemistry, same general biology, and the light-guiding structure is absent. The Pacific species fluoresces perfectly well without it. That contrast is the single most useful piece of evidence in the whole case, and it took me a while to see why. Think about what a waveguide implies. The tonic water in the fridge has quinine in it, and the quinine fluoresces. And nobody designed that. Fine. That's the byproduct model, and it's a strong one. But the bottle does not come with fiber optics molded into the glass to direct the glow outward. There's no plumbing. A byproduct gets to exist. It does not get infrastructure. So when you find a structure sitting on top of a fluorescent tissue that captures the emitted light and channels it, that starts to look like a system. Something in that animal's history appears to have been working on the light after it was produced. That's the argument. And it's the strongest one available. And I want to take it apart before I lean on it. Because it has a weakness that took me longer than it should have to spot. A denticle is a small, tapered, hard, partly translucent rod made of dentine and an enamel-like shell, sticking up out of the skin. Ask a physicist to design a light guide and they'll hand you a small, tapered, hard, partly translucent rod. Materials with a higher refractive index than their surroundings trap light inside themselves by total internal reflection. That's ordinary physics. And it happens any time light enters a dense, transparent solid, at the right angle. A glass rod does it. A strand of nylon fishing line does it. Run a flashlight into the end of a plastic drinking straw, and you'll see a version of it, which means a denticle might guide light for the same reason a wet spaghetti noodle does, because of what it's made of and what shape it is, with no selection for the optical outcome at any point in the animal's history. That's a real objection. It's the kind of objection that has quietly killed a lot of appealing adaptive stories in biology, and anybody making the signaling case has to answer it. Here's the answer, and it's partial. The chain cat shark has two kinds of denticle, smaller ones that guide the light and larger ones that don't. That's the detail that does the work. If the light guiding were a free consequence of being a hard tapered rod, you'd expect every denticle on the animal to do it, since they're all hard tapered rods. Finding two structural classes on one animal with different optical behavior means something has differentiated them. Two versions on one animal. That still doesn't get you all the way home, and I'd be overselling if I said it did. Denticles are differentiated across a shark's body for reasons unconnected to light. Different regions have different mechanical demands, different flow conditions, different exposure to abrasion. Two classes of denticle could exist for straightforwardly structural reasons, and the optical difference could be a passenger riding along on a mechanical adaptation, exactly the way the sexual dimorphism in the fluorescent pattern might be a passenger riding along on mating armor. Everywhere you push on this case, the same alternative is waiting, but notice that the alternative is getting more elaborate each time you invoke it. To hold the pure byproduct line now, you need a metabolic pathway that happens to produce green fluorophores, that happens to concentrate them in the pale patches and exclude them from the dark ones, in an animal that happens to have two denticle types where the smaller class happens to conduct the emitted light outward. All of it happening in an eye range the animal happens to be able to detect. At depths where the contrast happens to improve, every one of those is individually plausible as a coincidence. Stacked, they start costing something. That's not proof. Nobody should call it proof. It's the accumulation of unlikely alignments, which is how a lot of biology actually gets argued in practice, while everyone waits for the experiment that would settle it. Now, the part that bothers me about the whole comparison, and I haven't seen it raised anywhere, the species with the light guides is the deep one. Chain cat shark, out on the slope, in clean blue water, hundreds of feet down, where the ambient light is dim and monochromatic, and where the fluorescence contributes the largest share of the total. That's the animal with the extra optical hardware, the swell shark, in a kelp forest at 100 feet in green coastal water, where the fluorescent contribution is a smaller fraction of a brighter, greener background, doesn't have it. That lines up beautifully. The animal that needs the help has the help. And it's a sample size of two species, one comparison. That falls a long way short of a comparative analysis. It's an anecdote with a hypothesis attached, and treating it as evidence of a trend would be exactly the kind of reasoning this whole story should be teaching us to distrust, which is also, and I mean this sincerely, an opportunity sitting right there for somebody. There are well over 100 species of cat shark in the world. They live across an enormous range of depths, from shallow coastal water down into the true deep, in every ocean basin, in clear water and murky water and everything between. If the light-guiding denticles are part of a visual signaling system, there's a prediction that falls straight out of it. Deeper living cat shark species in bluer water should be more likely to have them. Shallow water species in green coastal conditions should be less likely to. The trait should track the light environment across the whole family. If it's a mechanical adaptation with an optical side effect, it should track something else entirely. Body size, maybe, substrate roughness, swimming behavior, mating style. That's a real study. Museum collections already hold the specimens. Somebody with a scanning electron microscope, a taxonomic key, in about 2 years could answer it. And the answer would either strengthen this case substantially or take a load-bearing wall out of it. Nobody has run it. As far as I can tell, nobody's proposed it in print. So the case stands where it stands. And it's worth going back now to that shark on the sand in the canyon off La Jolla, because we know a great deal more about what we were looking at than we did at the start. So, the shark in the crack, 100 feet down off La Jolla. Go back and watch it again, with everything we've picked up. The blue light falling on that animal has come down through 100 feet of coastal Pacific water that has already stripped out the red, the orange, and most of the yellow. What's left is the short end of the spectrum, arriving from above, weakened but plentiful. It lands on the shark's back. The pale patches on that back are loaded with small molecules that the animal built out of tryptophan, an amino acid it got from eating fish, brominated with an element it pulled from the seawater it's been breathing its whole life, and processed down a metabolic pathway so old that you and I are running the same one right now in our own tissues. Those molecules absorb the blue. They hold it for a few billionths of a second. They hand back green. Inches away, in the dark saddles, none of that is happening. Those patches take the blue and keep it. And a foot away, in the eyes of any other swell shark in that crevice, there are rods tuned to 484 nanometers, catching some fraction of that returned green, throwing it into the same bucket as everything else, and producing a brightness map in which the pale patches are a little brighter than they have any right to be, and the dark ones are as black as the rock. That's the scene. That was always the scene. The divers didn't cause it, and the lamps didn't cause it. We walked in on something that has been running continuously in that canyon since long before anybody was there to point a camera at it. The yellow glass bought us a seat at something already in progress. And that's the honest limit of that moment, which is why I don't want to leave the story there, even though it's the most satisfying place in the whole thing to stop. Because the dive showed us the sender. Every single thing in that scene is the shark's side of the transmission. Not one frame of it tells you whether anything is receiving. So let's go through the candidates properly, and I want to do this as a process of elimination, because several of the explanations that get floated in the coverage fall apart under about 90 seconds of pressure, and it's worth watching them fall. Candidate 1. Camouflage. The idea is that fluorescence could hide an animal instead of advertising it. If your background is fluorescing and you're fluorescing to match, you disappear in that channel the same way a green animal disappears in a green forest. This is not a silly hypothesis. Corals fluoresce heavily in a range of colors. Some algae fluoresce. A small cryptic reef fish sitting on a fluorescent coral head has a genuine reason to match it. And for reef species, that's a live and reasonable idea that the people who study fish fluorescence take seriously. Run it on our animals, though. The swell shark rests on rock and sand, and kelp hold fasts. Kelp does fluoresce, faintly, mostly from chlorophyll. And chlorophyll's emission is way out in the red, where the shark's eye barely functions, and where the water kills the signal within a few feet anyway. It isn't a green background. The chain cat shark is worse for the hypothesis. It lives on rough, rocky bottom hundreds of feet down. There's no coral field of the reef building, brightly fluorescent kind at that depth in that habitat. There's rock. Rock doesn't fluoresce green. So the animal is producing a color its background doesn't produce. That's the opposite of matching. Camouflage in the fluorescent channel requires a fluorescent background, and neither species has one. I'd call that hypothesis dead for these two species. It may well be right for a goby on a reef. It doesn't work here. Candidate 2. Luring prey. Glowing animal, curious fish, fish swims over, shark eats fish. It's an attractive story because it has a clean mechanism and because there's a spectacular real version of it in the ocean already. Deep-sea anglerfish. A modified dorsal fin spine extended out over the mouth, ending in a bulb packed with light-producing bacteria, dangled and twitched in the dark in front of a set of jaws. That's a lure. Everything about it says lure. It's isolated from the body, it's positioned relative to the mouth, it's under muscular control, and it's the only lit thing on the animal. Now look at a cat shark. The fluorescence is spread over the entire body: face, flanks, belly, back, fins, claspers. It's a pattern distributed across the whole animal in a fixed arrangement with no capacity to move it or aim it or switch it off. Since it runs on ambient light the animal doesn't control. Lures are localized and controllable. This is the opposite of both. And then there's the killer detail, which comes straight out of the swell shark's own natural history. A big part of what it eats is fish that are asleep, torpid, unresponsive, shut down for the night. That's the documented feeding specialization. You cannot lure a sleeping fish. There is nobody home to be lured. Two independent reasons, and I'd throw this hypothesis out without much regret. It survives in the popular coverage because it's easy to picture. Support is a separate matter. Candidate 3, anti-predator function. Startle, warning, confusion, something along those lines. This one doesn't fail. It just can't be evaluated. And the reason is stated plainly in the original research. Nobody knows what eats these sharks. That's the situation. The predators of the swell shark and the chain cat shark are essentially undocumented. There are reasonable guesses, larger sharks, some marine mammals for the shallow species, but no established predator with a known visual system that you could run the calculation against. An anti-predator hypothesis requires a predator. You need to know what it is, where it hunts, in what light, and what its eye can detect. Without those, the hypothesis is unfalsifiable, and an unfalsifiable hypothesis is a placeholder. I'll add one observation that pushes against it anyway. The swell shark's entire anti-predator strategy is documented, and it's mechanical. Wedge into a crack, inflate, become impossible to remove. That's a physical solution, and it's a good one. And an animal that has solved predation by turning itself into a cork has limited use for a visual warning display. Candidate 4. Photoprotection. The idea that the compounds are there to absorb damaging short-wavelength light and dump the energy harmlessly, with the visible green being incidental. This one is chemically respectable. Plenty of organisms do exactly that. Corals are thought to use some of their fluorescent proteins partly as sunscreen. It fails here on geography. Ultraviolet is gone from the water column at these depths. Long gone. And the swell shark, which lives shallow enough that UV could in principle reach it, spends the entire sunlit day jammed into a rock crevice and comes out at night. That's an animal with essentially no ultraviolet exposure, carrying what would have to be one of the least necessary sunscreens in the ocean. Dead. 4 hypotheses. 1 dead on background mismatch, 1 dead on anatomy and prey behavior, 1 unevaluable for lack of a predator, 1 dead on depth and daily routine. What's left standing is species and sex recognition, which is a suspicious way to arrive at a conclusion, and I know it because everything else failed, is the weakest possible form of argument. Elimination only works if you enumerated the possibilities correctly. And there is a 5th candidate that I've been holding back this entire time. It didn't come from vision science or from behavior. It came out of the chemistry, sideways, in an experiment that had nothing to do with light at all. And it's the one hypothesis on the list that could explain everything without a single shark ever looking at another shark. When a natural products chemist isolates a new compound from an organism, there's a thing they do almost reflexively, the way you'd check your pockets before leaving the house. They throw it at bacteria. It's cheap, it's fast, and the entire field of antibiotics came out of doing it often enough. So the Yale group took the new brominated compounds they'd pulled out of cat shark skin and ran them against bacterial cultures to see what happened. Several of them slowed the bacteria down. Not spectacularly, nobody found the next penicillin in a shark. But there was measurable inhibition, and it showed up against organisms that matter, including strains from the genera responsible for staph infections and for cholera. Real inhibition against real pathogens from a compound whose entire public identity is that it makes a shark look pretty. And once that result was on the table, a few other things clicked into place that had been sitting around looking like scenery. Start with the pathway. The kynurenine route, the one the shark is running its brominated tryptophan down, is tangled up with immune function in vertebrates generally. The first enzyme in that chain gets switched on during infection. Several of the intermediates modulate immune responses. It's a metabolic route with a long-standing relationship to defense against pathogens, and it's been in that role for a very long time. Then bromination. In marine organisms, brominated compounds are, overwhelmingly, defensive chemistry: sponges, algae, bryosins, sea slugs, soft-bodied things that can't run away and have to solve their problems chemically. Brominated compounds from the sea have been screened as antibiotics and antiphyllantes for decades. Bromine on a ring, in the ocean, usually means somebody is trying to poison somebody. And then there's the observation the researchers made about the animals themselves. These deepwater sharks are clean, no fouling, no visible microbial growth on the skin, in an environment that will colonize anything that holds still. Put those together, and you get a hypothesis with real teeth. The compounds are a chemical defense, a skin antibiotic, painted onto the surfaces most at risk, built from a common amino acid using an element the ocean supplies for free, running through a pathway that was already doing immune work before sharks existed. And the green is nothing. A leftover optical property of a molecule that's there for a completely different job: the tonic water in the bottle at full strength. I want to give this the strongest possible version because it deserves it, and because most of the coverage of these sharks skates past it in a sentence. Look at where the fluorescence is brightest. Now read that list as a map of infection risk: the claspers. Reproductive structures inserted, physically exposed, subject to abrasion during mating. If you were designing a topical antimicrobial for a shark and had to pick one structure to protect above all others, you would pick those: the ventral surface, the belly, the part of the animal that spends its entire life in contact with rock, sand, sediment, and whatever is living in it. Constant abrasion, constant exposure, constant microbial load. The face and head, leading edge of everything, into crevices, into the substrate, into prey. That's the same list twice. The regions that fluoresce most strongly are the regions most likely to get infected, which means every distributional fact I've been using as evidence for a signal is equally good evidence for a defense. Now push it into the dimorphism, and this is where it gets uncomfortable because I think this reading is stronger than anyone has given it credit for. Female cat sharks get bitten during mating. That's established. The male grips and holds with his teeth on the flanks, the pectoral region, the gill area. Females across several cat shark species carry heavier skin in those regions, and the standard interpretation of that armor is protection from exactly this. A bite breaks skin. Broken skin in seawater is an infection risk. So, what would you predict about the distribution of an antimicrobial compound in a female shark that gets bitten regularly compared to a male that doesn't? You'd predict more of it in the regions that take the damage, which is, as far as I can tell, precisely the observation: denser fluorescent spotting on the female's underside, a distinctive pattern on the female's head, sexual dimorphism in the distribution of a compound whose job might be wound defense in a species where one sex takes the wounds. I'll flag that clearly. That's my reasoning, not a published finding, and I haven't seen it laid out in the literature in those terms, but it follows directly from facts that are all individually established, and if it's right, then the single most persuasive piece of evidence For the signaling hypothesis, the sexual dimorphism evaporates. It becomes a fact about who gets bitten. That's the strongest rival explanation in this entire case, and I don't think it's been taken seriously enough. It has one enormous advantage over everything else on the table. It doesn't need a receiver. Every problem we've run into for the last hour has been a problem of perception. Can the eye detect it? Is the contrast sufficient? Does the behavior change? The antimicrobial hypothesis walks past all of that, because bacteria don't have opinions about photons. The compound works in a bucket. It works on a dead shark. It works in absolute darkness at midnight in a cave. Nothing has to see anything for it to be doing its job. So why don't I think it's the whole answer? One reason, the denticles. An antimicrobial compound has no use for a waveguide. Bacteria are not affected by which direction the light leaves the skin. If the entire point of these molecules is chemical defense, the optical behavior of the structures sitting on top of them is irrelevant. And there is no reason for the chain cat shark to have evolved two classes of denticle, where the smaller class conducts the emitted light outward along its axis. That one observation sits outside what chemical defense can explain. It's about photons going somewhere. And photons going somewhere only matters if something is at the other end. Which leads to the answer I think is most likely true, and it's the least dramatic one available. So it never makes headlines. Both. Evolution is relentlessly cheap. It does not build a new system when it can add a second job to an existing one. A compound that started as a chemical defense, present in the skin for reasons unrelated to vision, would be sitting there in the tissue with a bright green optical property going unused. Any animal that happened to benefit from being slightly more visible to its own species, in water where nothing else produces that color, would be under selection to enhance it, concentrate it in useful places, exclude it from others, and, eventually, reshape a structure sitting on the skin so that more of the light gets out in a useful direction. Defense first, signal second, built on top of it, using the same molecule. That's a recycling story, and recycling stories are the norm in biology. Your jaw contains bones that used to be gill supports. The bones in your middle ear that let you hear this were part of a reptilian jaw joint. Feathers were insulation, long before anything flew. Almost nothing in a body is doing the job it was originally built for. I like that answer. I'd bet on it, if somebody made me. But liking an answer and having evidence for it are different situations entirely, and it's time to be exact about which one we're in. In 2017, Justin Marshall and Sonke Johnson, two of the most respected people working on vision and light in the sea, published a review in the Philosophical Transactions of the Royal Society on fluorescence as a means of enhancing color signals. Their assessment of the field was blunt. Most observations of fluorescence, they concluded, lack enough evidence to suggest the fluorescence is being used in any visually driven behavior at all. They also put their finger on exactly the problem this whole story keeps running into. The central difficulty, they wrote, is telling the difference between a case where fluorescence is a functional feature of an animal's coloration and a case where it's a byproduct of a pigment or some other molecule that's present for other reasons. That review covers flowers, corals, worms, squid, spiders, mantis shrimp, fish, reptiles, parrots, and people. Fluorescence turns up everywhere once you start looking for it, and the number of cases where anybody has demonstrated that an animal's behavior actually changes in response to another animal's fluorescence is very close to zero. That's the field this shark lives in. There's a structural reason the field ended up like that, and it's worth naming because it explains a lot of the coverage you'll encounter about glowing animals. Fluorescence photography produces spectacular images. It's the most reliably beautiful thing in wildlife imaging. Point a blue lamp at almost anything with a yellow filter on the lens, and you'll get a picture that stops people scrolling. And that image is generated by equipment that no animal possesses. The filter removes the excitation light completely. Nothing in the ocean does that. Every animal down there is looking at the fluorescence and the ambient blue simultaneously mixed together, with an eye that in this case can't even separate the two by color. So the image that drives the excitement, the funding, the headlines, and often the hypothesis is an image of something no organism has ever experienced. That's a hell of a foundation to build an interpretive story on. And to their credit, the researchers who built the Shark Eye camera were trying to fix precisely that problem. Now, the specific gaps in this specific case, and I'd rather give you the list than paper over it, because the list is honest, and it's short enough to hold in your head. Gap 1, and it's the biggest, and the original paper says it out loud. There are no psychophysical models of shark lightness perception, which means that when the team calculated how much contrast the fluorescence provides to a cat shark's eye, they had to borrow the math from somewhere. And where they borrowed it from was human vision, human color space models, adapted to the shark's measured photoreceptor sensitivity, because nothing else exists. That's a legitimate and standard approach. It's also a proxy. Nobody has established how a cat shark's nervous system actually converts photon capture into perceived brightness, how much contrast it needs before a difference becomes detectable, or how it handles a background that's changing. All of that is unmeasured. The contrast numbers are physically reasonable estimates of the light arriving at the retina. What the animal actually notices is a separate question and an unmeasured one. Gap 2. Nobody knows what preys on these sharks, so anything involving predators can't be evaluated. Gap 3. The chain cat shark, which is the better test case on every physical ground, is the species almost nobody has observed alive in its own habitat. Most of what we know about its fluorescence comes from animals in tanks and tissue on benches. Nobody has taken a camera to 1,800 feet on the upper slope, waited, and watched what these animals do with each other under nothing but the light that's already there. Gap 4. Nobody knows whether the fluorescence changes. Think about how strange that gap is. If this is a mating signal, the most basic prediction in all of sexual selection is that it should intensify with sexual maturity, and quite possibly with the breeding season. Juveniles should be duller. Mature animals should be brighter. Nobody has measured a series of animals across age classes and reproductive states and looked. That's not an expensive study. It's a museum drawer, a blue lamp, a calibrated camera, and some patience. Gap 5. The comparison that generated the light-guiding Denical finding involved two species, and Gap 6, which sits underneath all the others. Nobody has ever run a behavioral experiment. Not one. In the decade since the first paper, no one has put a cat shark in front of another cat shark's fluorescence and measured whether anything at all changes in how it behaves. That's the state of the evidence. Everything we have is physics, chemistry, anatomy, and correlation. Every link in the chain is measured. The chain has never been connected to a behavior at either end. So what would settle it? The brief I set myself at the start was to say precisely that. And I think it comes down to two experiments, with 4 more that would help. The first one is a threshold measurement. Take a live cat shark in a controlled setup and determine the smallest brightness difference it can actually detect in the relevant part of the spectrum, at the light levels it experiences at depth. You can do this with electrical recordings from the retina, or with trained behavioral responses, or both. The output is a number, the contrast threshold. Then put that number next to the contrast the fluorescence actually generates. If the fluorescence produces a difference above threshold, the signal is at least detectable, and the whole hypothesis becomes live in a way it currently is not. If it falls below, the visual story is finished, permanently, and the antimicrobial explanation takes the field. One experiment. Binary outcome. Nobody has done it. The second one is a choice test, and the design matters enormously. Present a cat shark with two objects, same size, same shape, same movement, same everything, under natural spectrum blue illumination. One fluoresces, one doesn't. And here is the part that most versions of this experiment would get wrong. The non-fluorescent control has to return the same total amount of light to the shark's eye in the shark's own sensitivity band. If you just compare a glowing model against a dull one, you've tested whether sharks prefer brighter things, which we could have guessed. To test fluorescence specifically, the control has to be matched for total radiance as the shark's photoreceptors weigh it, and differ only in where along the spectrum that light sits. Then measure, approach, inspection time, orientation, any courtship behavior at all. If the sharks respond differently, you have your answer, and it's the answer everybody wants. If they don't, you've learned something equally important. Those two experiments would settle the central question. The other four would fill in the picture. Comparative work across the cat-shark family, testing whether light-guiding denticles track depth and water clarity. Seasonal and maturity sampling, testing whether the signal strengthens when it would need to. In situ observation at chain cat-shark depth under natural light with no lamp involved, and a straightforward chemical question nobody has answered. Does the concentration of these compounds shift with reproductive state? None of that is exotic. There's no new technology required. It's 4 to 6 years of unglamorous, careful work by people who would rather be doing something with more obvious funding appeal. Which is, honestly, why it hasn't happened. The photograph was the easy part, and it already went viral. And the hard part pays worse. So let me put the answer where the evidence actually leaves it, without rounding it up. Here's what we know, stated at the strength the evidence supports, and no higher. 2 species of small, secretive cat shark absorb blue light in the pale regions of their skin and re-emit it as green, peaking somewhere around 530 nanometers. The molecules doing it were unknown to science until 2019, a family of brominated compounds built from tryptophan and processed through the kynurenine pathway, present in the light patches and absent from the dark ones. In the chain cat shark, a subset of the dermal denticles conducts that emitted light outward along their own length. The compounds also slow the growth of bacteria. Both species have retinas made of rods, running a single visual pigment, at 484 and 488 nanometers, respectively. They cannot see color. They can absorb the emitted green as brightness, at a reduced efficiency, and physical modeling indicates the fluorescence increases the contrast between the animal and its background, and between the light and dark patches on its own body, with the effect growing more significant as depth increases and ambient light thins. The patterns differ between males and females. In both species, the male's claspers are among the most strongly fluorescent structures on the body. The leading interpretation, held by the people who did the work and by most of the field, is that this is species and sex recognition. A private visual channel in a monochrome blue world, used by animals that have specialized so hard in being invisible that they need a way to find each other. That interpretation is not settled, and the honest reason is simple. Every piece of evidence supporting it describes the sender. Nobody has ever demonstrated a receiver. There is a rival explanation that covers a great deal of the same ground. These are brominated compounds from a pathway with ancient ties to immune function, concentrated on the body surfaces most exposed to infection, in animals whose skin stays conspicuously clean in water that colonizes everything. They may be a chemical defense whose green glow does nothing for anybody. The two possibilities are not mutually exclusive, and I'd put my money on the answer being layered, a defensive molecule that acquired a second job. That's a guess with reasoning behind it, and it is a guess. 2 experiments would decide it. Measure the animal's actual contrast detection threshold and compare it against the contrast the fluorescence delivers. Then run a choice test with a control matched for brightness in the shark's own sensitivity band and watch whether behavior changes. Neither has been done. Both could be. That's the case. That's where it stands in the summer of 2026. Now let me tell you what I actually think this story is about, because it stopped being about sharks for me. Somewhere in the middle of the research, there were divers on that reef off La Jolla in the 1960s. There were divers there in the 80s, the 90s, the 2000s. Marine biologists, photographers, fisheries surveys, people who knew swell sharks intimately, who could tell you their diet and their breeding season and the exact crevices they favored. Every one of them swam directly past an animal that was converting blue light into green, continuously, right in front of their faces. Nobody saw it. Not because they weren't looking. They were looking hard. They saw everything their eyes and their equipment were built to deliver, and the phenomenon fell outside that entirely. It took a blue lamp and a piece of yellow glass. That's all. No submarine, no supercomputer, no genome. A lamp and a filter on a night dive that was about something else in 2011. And the moment somebody looked through the right piece of glass, an entire layer of the ocean turned on. More than 450 species of bony fish are now confirmed to do this, with dozens added in a single recent study. And the trait has been reconstructed as arising independently more than 100 separate times across the fishes, going back something like 112 million years to the ancestors of the eels. That number was 0 within living memory, in the sense that nobody had counted, nobody had looked, and the whole thing sat there being real and being ignored. We are still at the very beginning of that count. There are something on the order of 1,000 species of sharks, rays, and their relatives alive today. A handful have been checked. The 3 families where fluorescence has turned up so far were found more or less by accident, and every one of them lives a life built around concealment, which suggests the group of animals most likely to be doing this is precisely the group least likely to be examined. So the honest expectation is that the list is going to get much longer, and that a lot of what's on it is swimming around right now inside the range of ordinary scuba, in water people dive every weekend. That's what I can't get past. The gap between what's there and what we register is not a small gap. And it isn't closing on its own. It closes when somebody builds an instrument and points it at something familiar. Every time that's happened, the answer has been the same. There was more going on than we thought, and it had been going on the whole time, and the only thing that changed was us, which raises a question that has no answer yet and might not get one in my lifetime. What else is happening out there in a channel we don't filter for? Nothing mystical about it. Ordinary biology, running in plain sight, in some part of the physical world our equipment doesn't sample. Magnetic fields, pressure waves, polarized light, which a lot of marine animals can detect and we cannot, and which almost certainly carries information we have barely started reading. Chemical gradients we have no instruments delicate enough to map. There is no reason to think fluorescence was the last one. I find that thought enormously cheerful, personally. The idea that a well-studied reef, 100 feet down, 40 minutes from a major American city, still has whole layers nobody has looked at yet. That the frontier isn't necessarily far away. Sometimes it's just sideways. 2 things before I let you go, and both of them are actual requests. First, the comments, and I want a specific answer rather than a general one. 6 experiments came up along the way, and I told you which 2 I'd run first. I've changed my mind about that order twice while putting this together, and I'm not fully settled on it now. So, if somebody handed you a boat, a permit, and 4 years, which one would you start with? The retinal threshold measurement because it's binary and it either kills the hypothesis or opens it. The choice test because behavior is what everyone actually wants, or the comparative dentical survey across the whole cat-shark family because it's cheap, The specimens already exist in museum drawers, and it might quietly answer more than any of them. Make the case. I do go through them, and I've been talked out of a position in the comments before. Second, if you want more of this particular flavor, where the ending is a careful maybe, with the reasons attached and the evidence gets to disagree with the headline, subscribing is how I know that. It's a small thing, and it tells me exactly which kind of video to make next. Same with the like. Consider it a note that the honest version landed better than a tidier one would have. And here's where I'll leave you. Tonight, when it's dark on the West Coast, the sun will have been gone from that canyon for hours, and the swell sharks will unfold out of their crevices and go to work along the bottom. There won't be a diver within miles. No lamps, no filters, no cameras. The last of the daylight will still be filtering down through the water, and every one of those animals will still be taking it in, converting it, and handing it back out as green. For nobody, maybe, or for each other. We don't know yet. That's the good part.